talking about somatic mosaicism in development using long-read whole genome sequencing.
All right, hello everyone. I'd like to begin by thanking the conference organizers for this awesome opportunity to present my research to you today. Okay, so I hopefully don't have to define this too much. We all know that mosaicism is sort of this phenomenon that somatic and germ cells can each independently accumulate mutations from one another, such that in developed tissues we can see genotypically distinct subpopulations of cells across and between different tissues. Right, the factors that drive this mosaicism can be quite complex, one of the primary ones being timing, especially in the context of development. So in germ cells, we can have these germline mutations that are going to transmit directly to the zygote and theoretically be in every cell that derives from that zygote that harbors the mutation. Mutations can also occur later in development, occurring at the organogenesis stages so that they're secluded only to a particular tissue. Further, the mechanisms of mutagenesis are quite broad, contributed by endogenous and exogenous factors with unique impacts on DNA sequence composition. These mechanisms of mutagenesis can be detected with tools such as cosmic mutational signature analysis, which also indicates to us that the sequence context of where these mutations actually occur does matter and has distinct differences between different mutational signatures. When looking at the spacing of these de novo mutations, especially in the context of meiosis, we see that there's an elevated level of de novo insertions and deletions, as well as structural variants occurring at these meiotic breakpoints. This has been demonstrated in human germ cells, with these different plots demonstrating that there's an elevated occurrence of those different mutation types in and around those meiotic hotspots, or meiotic breakpoints, rather. Right. It's also been implicated in certain plant species that sites of heterozygosity may complicate this crossover in meiotic recombination. So specifically in the perennial peach tree, as well as Arabidopsis thaliana, some researchers have found that by crossing distinct cultivars or strains of these species and establishing a high degree of heterozygosity in the F1, and then selfing those F1 plants to create the F2 offspring, they see a relatively higher localized germline mutation rate in the F2. So an increased amount of mutations surrounding those sites of clusters of heterozygosity derived from the F1. Specifically, this increased the mutation rate by 1.8 in the peach tree and 3.5 times higher mutation rate in the Arabidopsis model. When considering the spatial context of these mutations, we know mutations can occur proximal as well as distal from one another. So in the proximal context, this really means how mutations can occur in a smaller intermutational distance from one another, with the distal meaning that they're occurring further from the previous mutation in a given chromosome. So these can be visualized with a rainfall plot, with the x-axis corresponding to a chromosome position, as well as looking more broadly, you can sort of zoom out and look at the whole genome. With the y-axis corresponding to that intermutation distance. So those dots, which correspond to different mutations that occur lower on the graph, are going to be proximal to one another or proximal to their previous mutation, with those occurring higher up being distal from one another. Across a chromosome, different regions can also be dense or sparse with mutations occurring in clusters and deserts. So sort of looking at this rainfall plot again, we can see that there's distinct regions where there's no mutations occurring or very distant from one another, which would be termed deserts, as well as clusters, where we can also see varying levels of clustering or levels of density of those intermutational distances. So you can also demonstrate this with a rainfall plot or rainbow plot by comparing different types of mutations in a rainfall plot. So these red dots here would correspond to CNVs, for example, where we can then measure the distance between an SNV and its next nearest CNV. With predictions of spatial associations by looking at those sort of dips in the plotting on a rainbow plot. Okay, so taking this all together and considering the spatial relationships between heterozygosity and de novo mutagenesis in the F2, we began looking in our lab at outbred mice and seeing if there's any associations between those de novo CNVs and those heterozygous sites. We did this with a MDGA genotyping mouse array and had looked at outbred strains, so specifically the CD1 and NMRI strains, where we observed prominent spatial associations, proximal associations between heterozygosity and the incidence of de novo CNVs. However, when considering the sort of limited scope of genotyping arrays, I decided to extend this with long-read sequencing, so being able to detect all types of mutations with that unmatched phasing capabilities. I kind of did a breeding schema set up with the Jackson labs that is comparable to the plant studies, where I bred the B6 and DBA strains, which are distinct strains of those mouse species, and got the F1 with a relatively high level of heterozygosity in this inbred cross. By mating the brother and sister of the F1, generating those F2s, I then wanted to essentially, again, test the relationship between those F1 heterozygocytes and the F2 de novo structural variants more broadly. I did this by collecting high molecular weight DNA from 3 different tissues, specifically looking at the cerebellum, heart, and liver of all of these different mice and comparing them in this intergenerational approach to elucidate those de novo mutations. I chose these different tissues to sort of serve as a proxy for the early zygote, considering that these all come from different developmental germ layers, with the thinking that if a mutation is present in all 3 tissues, then it must have been present in the zygote, or at least the early zygotic stages, so sort of an inference of what germline variants would be present in the F2. Following long-read sequencing data, I sort of compared the mutation profiles between the different tissues, as well as elucidating those germline variants, where I observed, prominent tissue-specific mutations that are occurring in an individual mouse. So these would be those post-zygotic mutations that would be, consistent with the developmental distinct timelines of these different tissues arising in the mouse. The detection of those putative germline variants demonstrated that there was a range of around 49 to 65 de novo structural variants in the F2s, which again are present in all 3 tissues and are sort of an approximation of the germline mutagenesis. I then used J-statistic tools, which are a tool developed in the lab that can actually test whether there's a statistically relevant relationship between the spacing here, testing these different 4 possible scenarios where you could have distal relationships, meaning that this de novo structural variant is occurring further from clusters of heterozygosity. You could have no association, where it looks sort of random between the spacing here. You could have proximal, meaning that the SVs occur within or close to those highly dense regions of heterozygosity, or both, where they could occur within as well as distal from different clusters of heterozygosity. So these are mapped on the J-statistics sort of function map, where the value of the J function, as calculated through here, sort of corresponds to these different mutational patterns. If it's above this random distribution, that would indicate to us that there's a distal relationship. If they're sort of underneath that random distribution, they would be considered proximal to one another. You can also have no association, where this J function sort of maps in between those confidence bands, indicating just a random distribution of SVs to those inherited F1 heterozygosity sites, as well as both, so dipping under and above that sort of proximal and distal regions. So I did just this for the F1 and to F2 comparisons, looking at a chromosome by chromosome basis. So although I only had 3 F2 tissues, or 3 F2 mice, and then 2 F1s, doing this in this sort of chromosome pairwise comparison yielded 120 possible tests of this hypothesis of proximity of the de novo structural variants. So I'll just show a couple of results for some of the plots, and then sort of show a broad level of what we found. So I did observe, non-random associations in terms of the proximal context. This plot demonstrating the F1 father, I did mislabel that plot, so it is the F1 father compared to one of the F2 mice, specifically on chromosome 8, where I observed prominent proximity between the occurrence of, again, those de novo structural variants and the inherited clusters of heterozygosity observed in the F1. I also saw distal regions in the chromosome 2 comparing F1 to F2. As well as both. So again, dipping in and out of those proximal and distal regions. Overall, around 51% of these non-random associations were observed to be proximal, as well as 22% being both. So really showing the complexity of the spacing though demonstrating that this was significantly more abundant than expected at random of the proximal associations, with evidence that corroborates this hypothesis of heterozygosity that associates with elevated local germline structural variation. The next steps would really include detecting germline variants directly in the outbred mouse models. So I'll be looking at a range or spectrum of differing levels of heterozygosity clustering in the genome and seeing how that relates to de novo structural variants. As well, I hope this presentation, though I briefly just touched on the J statistic, kind of shows that this tool can be used with your own data to measure different spatial relationships between two distinct genomic features, which could include different mutation types. All right, so with that, I'd like to acknowledge my supervisor, Dr. Kathleen Hill, as well as current and former students of the lab who have really contributed to this work, as well as our funding, and again to the Wellcome organizers for giving me this opportunity to present. Thank you.
Okay, questions? One here. Thank you.
Nice talk. Do you know about sort of a more on the topology of some of these heterozygosity association with this de novo CNV? Do you know much about it?
Yeah, I didn't have time to include the plots, but I did look at the various sort of densities of heterozygosity across the genome. So when doing this J statistic, it came to sort of a minimum threshold for what I considered a dense site of heterozygosity, and then sort of used that as the baseline for detecting these clusters.
And does it associate with any certain sort of phenotype or characteristic in the genome, like methylation dense or any other marks that?
Yeah, I haven't, with the PAC biosequencing, we do get the methylation marker, so we could integrate that data. I haven't quite yet, but.
Nice, thank you.
I think one.
So do you have any idea about the mechanism of what's driving up these structural variants in the context of the diversity?
Yeah, we're thinking that it's due to non-allelic homologous recombination. So one of the previous students in our lab investigated more in depth with those CNVs and the F1 heterozygosity sites. And we're thinking that it causes some sort of strand slippage during meiotic recombination and sort of increases the amount of CNVs and structural variants more broadly that are occurring. This research more providing the evidence that there is some level of de novo germline mutagenesis occurring locally, and we haven't exactly parsed out the mechanism quite yet, but we're thinking it's that NAHR.
And do you see elevated point substitutions as well, or just the copy number?
Yeah, we did detect the SNVs and indels for the F2 as well, and I just didn't test it quite yet, so I've really just been focusing on the structural variants given the prior data that found those elevated CNVs.
So related question. The numbers you showed of de novo, I guess, variants, that was structural variants?
Yes.
Per se?
Yeah.
Okay. And has anything like that been seen before in mice? I guess people didn't really have the right technology, but is that number surprising? Because this is quite high. Or do we know anything from other species about structural variants and de novo acquisition in normal tissues?
Well, I think we need to compare different generations as well in that same family. So looking at the de novo rate from the F1 to the parents, which I didn't quite look at, to sort of see if that's normal, if it's elevated in the F2s. And then with the number, because it's a sort of approximation of germline variants, it could actually be much lower. And those could just be early post-zygotic stages that were pre-cleavage of the germ layers. So it could be, inflated by the early zygotic stages.
Are you doing that across strains? Because you select a couple of strains, right?
Yeah, I started with the inbred cross, but I'm going to be working on CD1 mice, and then which are an outbred strain, and then maybe some other outbred strains as well.
Okay. Awesome.
Hi, Alexis Gerritsen from the University of Utah. Really nice. I was wondering, BXD and are mice recombinant inbred panels that exist, and I was wondering whether, one, you recapitulate the signal of the MutYH mutator locus that's present in DBA, and also whether you've looked to see whether the mutations that accumulated during the BXD breeding funnel follow this sort of pattern that you've observed here, since there's many to go on across the two genomes.
I haven't done any comparisons yet to the BXD. I did find those, and I was kind of looking for a sort of assembly for those genomes, and given how broad the BXD panels are, I couldn't exactly find a reference genome to compare it to, but I think that would be definitely worthwhile in terms of comparing and seeing how those my cross would sort of relate to the sort of established crosses that are present.
Okay, thank you very much.
So, thank you.